All right, let's move on to objective 3 and let's get into more of antidiuretic hormones. So ADH, we kind of briefly introduced this idea of how it acts. We're going to talk more about the triggers or the states that will help to release this antidiuretic hormone, and then more about how it acts in terms of that biochemical mechanism. All right. So the effects of ADH on water reabsorption are basically helping to change the permeability of the distal tubule and the collecting duct to water. So it's basically going to help, as we mentioned before, insert those aquaporin 2 channels and determine our urine osmolarity. If we insert more of those aquaporin 2 channels, we can reabsorb more water, and we're going to have a very low urine osmolarity, around 100 or so. If we have less of those aquaporin 2 channels, because we have less ADH, we're going to insert less of those into the membrane, and we're going to have less water being reabsorbed, and we're going to have more. Wait, I think I flip-flopped that around. So if we reinsert more of the channels, we reabsorb more water, we're going to have a more concentrated urine, okay? If we can reabsorb or reclaim more water, we have more solute and less water in terms of our filtrate, in terms of our urine, and that is going to give us an osmolarity of about 1400, okay? So more ADH, more aquaporin 2 channels, an osmolarity of about 1400. On the other end of that spectrum, right? If we're well hydrated, we have less ADH in our system, we insert less of those aquaporin 2 channels, we reabsorb less of that water from our urine, from our filtrate. And so we're going to have a more dilute urine, which has a lower osmolarity of about 100. All right. So ADH, if you think about the name, antidiuretic, it's preventing diuresis. It's preventing water loss, right? It's going to help us to reabsorb more of that water, especially in times when the body's in a dehydrated state, blood pressure and blood volume are low, as a way to help maintain that blood volume by reabsorbing that water. Okay. All right. So let's talk about the mechanism by which that is accomplished. So again, we have ADH being secreted into our plasma. And just to kind of go through the backstory of where this ADH is coming from, if we remember our pituitary from last semester, it had an anterior part and a posterior part. The posterior part is what was releasing ADH, right? This is also called vasopressin or antidiuretic hormone. And so the posterior part of the pituitary, if you remember from our discussion before, is releasing this hormone. So it's not actually made in the posterior pituitary, it's made in the hypothalamus, but because of those neurosecretory neurons, it's going to be communicated down into the posterior pituitary, and then depending on our hydration status, our blood volume, detected by certain receptors that we'll talk about in a minute, we're going to secrete this ADH into the blood as a hormone, right? Now that is going to make its way all the way to the kidneys, and it's going to bind to its receptor on the principal cells of the distal tubule and the collecting duct. So the very specialized cells that are found at the distal tubule and the collecting duct are called principal cells. And this is where this unique receptors that will bind this ADH are located. So we've got these ADH receptors, ADH circulating in the plasma can bind here to this receptor. And this is a metabotropic receptor. So it's going to follow the similar principles that we described for these slow or metabotropic receptors. It's going to be coupled to a G protein. That is going to have a change in its shape such that one of the subunits dissociates off. It's going to activate adenylate cyclase, which is our amplifier enzyme, and that is going to help to secrete or generate our second messenger. So adenylate cyclase creates cyclic AMP, which is our second messenger. That will then go into the cell and bring about our amplified response, right? That will help to activate protein kinase A, and that will help to generate or to make and to insert these aquaporin 2 channels. Now notice the aquaporin 2s are the ones that we can upregulate or downregulate by inserting more or less of those into the apical surface of the membrane. Aquaporin 3 is always present in the basolateral surface. So we don't really upregulate or downregulate aquaporin 3. It's always present as a way for that water to go from the basolateral surface across the interstitial fluid and into the peritubular capillary. Okay, so this doesn't change, but the aquaporin 2s, which are on the apical surface, are really what ADH manipulates. So because of the binding of ADH here, we secrete more of these aquaporin 2 channels, or we make more of them rather, and we insert them into the apical membrane. And that is how we can facilitate the reabsorption of water from our tubular space. So here in the tubular structure, which is the distal tubule and the collecting duct, and so we can pull all the water from our filtrate and reabsorb it into the cell, and then through this aquaporin 2 across the basolateral surface and into our peritubular capillary. And this is how we reclaim water from the tubular structures, because it would otherwise be lost in the urine, right? And because we don't want that, we help to reabsorb it back into the vasculature. Okay. Right. Kind of jumped ahead of myself there. All right, so that is the function of ADH, and talk about how the regulation of ADH, how it's increased or decreased. So ADH regulation happens via two mechanisms. First of all, is released from the terminals of the posterior pituitary cells. So this is basically the cells that are starting up in the hypothalamus. This is where the cell bodies of those neurons are located, and their axon terminals extend all the way down into the posterior pituitary. Okay, and then that ADH can be released from the posterior pituitary, although it's actually being made or secreted up in the hypothalamus. That is released in response to two signals or two states. First is the osmoreceptor. So the osmoreceptors that are in the organum vasculosum or the laminae terminalis, really fancy terminology there, sometimes known as OVLT, also known as VOLT. So you may read this up in other literature that says VOLT. So organum vasculosum actually translates to vascular organ. So vascular organ of the laminae terminalis. So a couple different terms there for that structure. It's basically a part of the brain that is closely associated with the third ventricle. So right around the third ventricle, this part of the brain does not have a blood-brain barrier, so really important to understand how it functions. It's not separated from the vasculature, so it's in direct connection or association with our plasma, with our blood, because of the absence of a blood-brain barrier. And so it's basically detecting or sensing the osmolarity of our plasma. It's going to then communicate to other areas, such as the thirst centers. So if the plasma is really high in osmolarity, it's going to help secrete that. If the plasma is really high in osmolarity, it's going to help secrete ADH as a way to reserve or reabsorb more of that fluid. It's also going to send signals to our thirst centers to make us more thirsty, as a way to bring in more fluid as well. Okay, so it is not surrounded by a blood-brain barrier, so that it comes in direct contact with our plasma, and it will detect the osmolarity of our plasma. And again, just reiterating the definition of osmolarity here, how much solutes is in that plasma? How salty, how concentrated is the plasma versus how much fluid or water is in that plasma? So if it has a high osmolarity, it's got little fluid and more solutes. And so that is not ideal. And so we want to think about ways that we can start bringing more fluid in by secreting ADH to reabsorb more of that water, or sending signals to our thirst centers to help us ingest more water through the diet. The second trigger or signal for ADH release is baroreceptors. So baroreceptors are detecting a similar phenomenon. They're detecting blood volume and blood pressure, and they're also going to secrete ADH in response to changes in blood volume and blood pressure as well, all right? So if we have low blood volume and low blood pressure, which means there's little fluid, little volume in our plasma, in our circulatory system, this is another trigger to secrete ADH. So if there's a little stretch being perceived by those baroreceptors because of low blood volume, they're going to send signals to the posterior pituitary to have that ADH released. And ADH will go to the kidneys by the mechanisms that we just described, help to reabsorb more fluid, and that will help to increase our plasma volume and therefore increase our blood pressure. Okay. All right, so let's walk through a few scenarios here to kind of illustrate those two examples. So these are the two scenarios or triggers by which we secrete ADH, or the two stimuli for ADH secretion. So the first one is increased osmolarity of our extracellular fluid or our plasma. And that is detected by the osmoreceptors in that region of the brain, the OVLT. And they are then going to increase the activity of the neurosecretory cells in the hypothalamus and allow for more ADH secretion from the posterior pituitary. Okay, so it's really important that you understand that anatomy, that the signals are coming into the hypothalamus where the cell bodies of these neurons are, but the actual ADH is being secreted or released from the posterior pituitary where the axon endings of these neurons are. So when we have that increased signal in the hypothalamus, that results in increased ADH from the posterior pituitary, and that will go to the kidneys, bind to that receptor, insert more aquaporin 2 channels into that apical surface, reabsorb more water, so increase water reabsorption in the kidneys via that mechanism, decrease water excretion, so we're now losing less water, which is what we want if we're in this dehydrated or high plasma osmolarity state. And that will help to conserve body water, and that will then turn off this initial error signal, okay? So some clinical or practical examples of this are, again, excessive exercise where you're dehydrated, excessive sweating, hemorrhaging, things of that nature, which will make the plasma osmolarity extremely high. And so that will trigger this sequence of events, which will allow for ADH to reabsorb more water and help to correct that initial error. Okay. The second stimuli for ADH secretion was the baroreceptors detecting blood pressure and blood volume. So again, if we have similar types of clinical scenarios, low blood volume, hemorrhaging, dehydration, the initial detector is going to be those baroreceptors that are detecting how much stretch exists in our vessels. And that will then send signals to those regions again in the hypothalamus, which will then result in ADH secretion from the posterior pituitary. That will result in water reabsorption from the kidneys via the mechanism we described, and that will decrease water excretion. So we're now holding on to more water, conserving more of that fluid, more of that blood volume, and correcting our initial error signal, okay? So if someone got stabbed, right, they're bleeding out, this is the type of mechanism that we're going to see as a way to preserve blood volume, right, to keep fluid levels up, to keep blood pressure up by preserving more blood volume. Okay. I'll take another pause here and see how we're doing with that so far. So someone had questions about ADH. If you still have questions, feel free to raise those now. So ADH is a really important clinical hormone. It's given exogenously after any type of trauma. Even after surgery, if there's extreme blood loss, we give ADH right away as a way to improve blood pressure by improving blood volume by the mechanisms that we just described. Okay, let's talk a little bit here about GFR and regulating GFR. So we began this conversation, we talked about the fact that GFR is very tightly regulated. We like GFR to stay at 125 milliliters per minute, even in the face of changing blood pressure. That is, if blood pressure is between 80 and 180. So if blood pressure is between 80 and 180, GFR is 125, and because of the intrinsic mechanisms that we described in a previous packet, we thrive to keep GFR at 125, even though we have slight fluctuations of blood pressure. Now, if blood pressure drops below 80, we have no choice but to drop GFR. So if we're in a scenario, again, bleeding out, dehydration, and our blood pressure drops below 80 millimeters of mercury, then we can no longer sustain GFR at 125, and GFR will in fact decrease, which will allow for us to compensate for that drop in blood pressure. So when GFR decreases, we have less water being filtered across the glomerulus, so less of that fluid being filtered from our plasma initially, and then we have less water being excreted. And so this is a way to conserve fluid loss in the face of a drop in mean arterial pressure or a drop in our blood pressure. All right. So this is to say that we're not just going to keep GFR, we're not just going to keep filtering the same amount of fluid if blood pressure drops dangerously low. So if it's below 80, this is a time to say, okay, we got to drop GFR as a way to compensate, hold on to more of that fluid as a way to help preserve or reserve that blood volume. On the other end of the spectrum, if we have high blood pressure, right, sustained blood pressure, if our blood pressure is above 180 millimeters of mercury, this is another time where we are not going to keep GFR the same. GFR will in fact increase as a way to protect the kidneys, protect the body, to lose some of that fluid. So we see an increase in GFR if mean arterial pressure or blood pressure is above 180. This results in an increased amount of water being filtered across the glomerulus and an increased amount of water being excreted or lost as urine, okay? Now, this only occurs in pathological circumstances. So think about really high, sustained, chronic hypertension. This is why hypertension can be so dangerous, especially if it's sustained and chronic, because it exposes the kidneys to this really high pressures and they dial in a new set point. So they're going to think that 180 is the new normal, and they're going to thrive to maintain that 180. It's also going to be damaging to the glomerulus. So one of the symptoms of kidney failure and kidney damage due to sustained hypertension is blood in the urine. And because of the chronically high GFR and chronically sustained higher blood pressures, we actually damage the glomerulus, damage the afferent arteriole, damage the efferent arteriole, and we end up seeing that spillage of blood into the urine, which again is very dangerous in terms of a sign of kidney dysfunction and kidney failure.